the replication, the complexity, whatever I told you, that's really complex. But the scientist who devised this technique, this is called polymerase chain reaction, is much more simple. And you don't need so many things like helicase, like primase, topoisomerase, nothing you need. You need just one enzyme, that is DNA polymerase. And that's why we named this technique as polymerase chain reaction. Just polymerase is good enough. Whatever I mentioned, so many enzymes, so much complexities, that is true for what is happening inside our cell, inside the living system. But if you are going to just replicate DNA or synthesize DNA inside tube, nothing is required. It's a very simple process. So let me go through it. So the PCR is an exponentially progressing synthesis of defined target DNA sequence in vitro. As I'm telling you again and again, that this is in vitro process. In vitro means inside tube. In the lab, you are making this, not in the cell, inside the cell. And in 1983, Kary Mullis, so he discovered the polymerase chain reaction, this process, and that's why he has been awarded with Nobel Prize also in 1993. So you can see almost all the steps, all the discovery, they are so important in biomedical science that they got Nobel Prize for almost each discovery we are actually explaining here. Either they got Nobel Prize in chemistry or they got Nobel Prize in physiology and medicine. So for example, Kornberg, he got Nobel Prize in chemistry. So on and so forth, almost all these techniques and the discovery. And now, why polymerase? I already told you, you don't need so many enzymes, just polymerase, that is good enough. And the enzyme specifically used in this reaction is called Taq polymerase. The name of this enzyme is Taq polymerase. So DNA polymerase, it's a DNA polymerase, but name is particularly Taq polymerase. This name came from one bacteria that is called Thermus aquaticus. So Taq, this capital T is this T and AQ is this, Thermus aquaticus. And the why chain? It is called chain because the products of the first reaction become substrate of the following one and so on. So if you have just this small DNA in the next round, so what you will have? So this is the strand plus strand, right? The next round, again you will get so exponentially it will be synthesized because all those stand in every round, they are acting as substrate and you will be getting all those things. Now, what are the components you need in order to carry out polymerase chain reaction? If you understand the replication, then you can provide the answer of this question. So what are the components you need to carry out PCR reaction? Tell me. Just one of you, just unmute yourself and try to answer. What are the reagent you need to carry out polymerase chain reaction? Who will tell? Just I would like to see that how much you can retain. Like just one of you, just please unmute yourself, and if you cannot answer that, nothing. But I'm sure that you will be able to tell what are the reagent you need to carry out the reaction inside you. Tell me. Aryan? You are here, so I can see.
Sir,
Yes.
Mg2 plus I remember.
Yeah, okay. You told one. Aryan told one. So Sukanti, Sukanti. So what about you? Can you try? So I just got one, that Mg2 plus. But Aryan, that's good that you quickly unmute and you told like Mg2 plus. But that's that ion, divalent cation. But apart from that, you need many things, right? Like while I was explaining the replication. So Avilas,
DNA templates.
DNA template. So you need a defined target first, right? So that one only you are trying to amplify. That's very good. So I got two. Then the next one,
Primase.
prime,
Primers.
prime,
RNA primers.
RNA primer. Very good, excellent. RNA primer. But since it is in vitro, in the in vivo process, what happened that there are a lot of problem actually to get the the initial the DNA polymerase. They need existing 3 prime OH. That's why a lot of complexity. But in the tube, we are trying to carry out this reaction. So one reagent should be DNA primer, not RNA primer in this case, because we are doing it in vitro. It's in our control, right? So DNA template, magnesium ion, and we will add DNA primer. That means small stretch of small DNA that will anneal, that will bind to the template DNA. If it is small segment, you have the 3' OH free. Now DNA polymerase will extend it, right? So that's all. So 3 things done. So what else? What else required?
Triphosphates.
Very good. Triphosphate. That means all 4 nucleotides, right? A, T, G, C. You need that. Very good. Then one big thing is still missing. So
Polymerase.
polymerase, exactly. Polymerase, DNA polymerase. So now what is happening? This is very interesting. So you have double-stranded DNA, right? Say, just for example, I'm telling you that you have just one DNA molecule at the beginning, and I'm just telling you this is the tube. It's not a tube, a cup, but it's a tube. You have DNA molecule, double-stranded DNA, and inside the tube, whatever you guys told, all those things are there. Magnesium chloride as the source of magnesium ion, DNA polymerase, DNA primer. Primer means small segment of nucleotides that will bind to template DNA and then it will be extended. DNA polymerase, everything there. Now we have to start the reaction. So then remember, while I was explaining in case of replication inside the cell, topoisomerase, helicase, primase, someone actually told right now that primer is required, RNA primer. You are right, based on your previous knowledge about whatever I discussed today. But in this case, nothing, because it is in the tube. This tube, we will just hit this tube. We will just increase the temperature of the tube. That's all, right? So if you increase the temperature of this tube, then what will happen? You learned during last week that whatever the double-stranded template DNA you have, you are trying to amplify that. That DNA will be in the single-stranded form because you are increasing the temperature, right? So as a result of that, without having helicase, topoisomerase, anything, you can just separate the 2 strands present in this tube. That's all. Now, you might ask that you need almost 80 to 90 degree temperature in order to separate the DNA strand. Then how we are going to do this? Because at this high temperature, what will be the status of DNA polymerase? Right? That's the question. Because in our body, temperature is fixed. It's ambient temperature. But that's the beauty of this polymerase chain reaction, and that's why it's a very clever and simple technique. although we are raising the temperature of that tube up to 90 degree to 95 degree centigrade temperature to separate the DNA strand, right? But enzyme will be fine, no problem. Those scientists cleverly, they have isolated, they have taken this enzyme from bacteria. That bacteria, I already mentioned its name, Thermus aquaticus. And this is called Taq DNA polymerase. Do you know where Thermus aquaticus live? They live in hot spring. You should know that there are many organisms, they can stay in sub-zero temperature in Arctic region, as well as many organisms, they can sustain, they can be present in almost boiling temperature. For example, hot spring, right? Particularly this bacteria, Thermus aquaticus, they stay in hot spring. So then what's the property of this enzyme? They are still surviving at that temperature, right? So they have some additional mechanism that help them to survive even at very high temperature. So the beauty of this enzyme is this enzyme will not be destroyed even at 90 degree, 95 degree temperature. It will retain its enzymatic activity. But our enzyme, our DNA polymerase, topoisomerase, nothing. They will be completely destroyed at this high temperature. So as a result of that, just having this enzyme that enable us to increase the temperature. So as a result of that, strands are separated. Then you are cooling down the temperature of the tube. So while you are cooling down the temperature of the tube, then the primer will anneal, bind to the template sequence. And at optimum temperature, around 70 degree temperature, this enzyme, they can work very nicely. So they can start extending or they will synthesize the DNA. Nothing you need. You have the DNA primer and it will be extended by the enzyme in presence of magnesium ion. And you already added all these 4 DNTPs, right? All nucleotides. And then after this round, what will happen? From one DNA molecule, you have now 2 double-stranded DNA. Then what will be the next step? Logically tell me. Whoever trying to attending that class, then what will be the next step? So we synthesize from one double-stranded DNA. Now you have 2 double-stranded DNA after this round. Then what will be the next step? Tell me. Avilas?
2 to 4.
2 to 4. But how you will do that? What I need to do?
Semi-conservative application.
Huh? Tell me. So we were hitting the tube and we separated both the strand. Then we are lowering down the temperature so that those DNA primer can anneal. Anneal means it can recognize particular sequence of the DNA. And then the primer is being extended, the DNA primer. This is not RNA primer. You don't need to think about removing those things. So now you have double-stranded DNA. So the next step will be, again, what you have to do? You have to increase the temperature of the tube because you have now 2 double-stranded DNA. Again, you have to make it single strand so that next round of synthesis will be happening because you have added a lot of primers, like numerous primer. Those are small, stretch of DNA, small DNA. You have added that, a specific sequence of primer that can bind to this defined target DNA. So as a result of that, in the next round, again, you will increase the temperature. So all those 2 DNA molecule, double-stranded DNA, they will be separated. That means all those strands now in single-stranded form. You have 4 strands now. So as a result of that, again, after high temperature, then again lower down the temperature. Again, primer will bind and it will be extended. So now you have 4 double-stranded DNA. Let's see what is present in the slide. Then I hope that it will be a little bit better. So target DNA, which we are going to amplify, pair of primers, you need 2 primers. Because see, why you need 2 primers? In the next slide, you will see. And DNTPs, thermostable DNA polymerase, and the magnesium as cofactor, and buffer, just the pH, salt, all those balance should be there. Now, this is the PCR reaction. If I go to this slide very slowly, and you should just try to concentrate here, then you will understand. So this is what a double-stranded DNA molecule. And the reactions are mentioned here, 5' to 3' and 3' to 5', those 2 strands. And this region you are interested to amplify, you are making you want to amplify that portion. In the first step, as I mentioned, if you hit whatever in the previous slide I mentioned, all those reagents, those are present in a small tube, and now you hit to separate both the strands. The hydrogen bonds will be broken and those 2 strands are getting separated. And now what will happen? Now you will see that if you cool little bit, you have to lower down the temperature. So as a result of that, the primer, the small segment of DNA, that will now bind. So that means you have to add specific primer so that it can recognize or it can bind to this DNA molecule. And now primer will bound here. So as a result, will be attached here. So for example, this is 5', this is 3'. See why 5', 3'? So this is 5', 3', 3', 5'. I told you always that DNA can be grown only from 5' to 3' direction. So 5', this one, it can grow in this direction. And here, this primer bound in this direction, 5', it will grow in this direction, right? So that's why you need 2 primers. One is called forward primer, another one is called reverse primer. You can forget about this concept of forward and reverse now, but in order to make this PCR happen, you need both the primers. Now, what will happen? It will be synthesized because in the solution you have DNA polymerase, you have all these 4 nucleotides A, G, C, T. So as a result of that, new strand being synthesized. So again, you have double-stranded DNA here, 1 DNA, another double-stranded DNA here, 2 DNA. So this is just one cycle of the PCR reaction or polymerase chain reaction. But I told you this is a chain reaction, polymerase chain reaction. So in the next cycle, what will happen? Next cycle, you will see this DNA molecule, right? So this another one, 2 DNA molecule, 1, 2, here, 1, 2. Similarly, again, you have to heat and you have to cool down, and then you will get 4 DNA molecule. Exponentially, it is increasing. Then again, you can repeat the step and you can continue for multiple cycles. Generally, we set the cycle around 30 to 35 cycles. So tell me, one of you, please unmute yourself. And if I tell you that in one tube, you started with 1 DNA molecule, right? So after 30 cycle, theoretically, how many DNA molecules should be there? After 30 cycle, how many DNA should be there?
Super 10 to 9.
A billion.
Yeah, billion. Yeah, I wanted to ask this to the power 30, 29, whatever you were saying that, because it's theoretically, right, it's exponentially growing, right? So as a result of it's definitely it's more than a billion. So that's why this is a very powerful technique, right? So as a result of that, you can synthesize almost within like 1 or 2 hours. You can make huge amount of DNA very quickly. So these are the small tubes we use in the lab. This is very common technique. Again, we use this in lab in a routine basis. So these are the small tube. It holds around 100 microliter of sample, small tubes actually. And this is the machine. This machine is nothing. This is thermo circular. So what's the purpose of this thing? You can put those tube, reaction mixture present here, DNA, all those things, whatever I mentioned. You have small hole here. Okay, you have to put those tubes here, and this machine can ramp up the temperature very quickly. It can go up and down very quickly. It can do that thing. So that way it can accelerate the reaction. But when people actually discovered this technique, that time this machine was not there. You don't need to have the machine. Just, you can set water bath at different temperature, like some water with different temperature, and you can just put the tube. Only the system is not efficient, but scientifically that is correct. And this is technologically improved now what we use this kind of machine, and very quickly it can and very accurately it can go up and down with the temperature. And when the reaction will be done, then tell me how you will check whether the DNA amplification happened or not. Tell me. You guys are I believe you guys are very good students. Tell me that after the reaction, how you will ensure that your DNA amplification happened or not. Just one of you try to answer this question, that the reaction you set in the machine for 2 hours is done, but how will ensure that it has happened, the reaction is successful? Based on whatever you have learned, you should be able to provide this answer. How you will test that?
Higher intensity of the UV light.
UV light, yeah, UV light means you are selling the. I hope that Anurag, so you are trying to say that you want to carry out gel electrophoresis, right?
Yes, after
yeah.
Measure the
Exactly.
intensity.
Exactly. So that's the answer. So whatever I discussed before, the gel electrophoresis, you have to do that, because at the beginning, the polymerase chain reaction, I added only few DNA molecule. Just I started with one DNA molecule or maybe 10 DNA molecule. So that many, like 1 or 10 DNA molecule, whatever you started, even 100 DNA molecule, that is not possible to see that just 10 DNA molecules just in the gel electrophoresis. I understand because of your experience, like you are not able to telling this thing. Maybe you thought that just one DNA molecule also you will be able to see in DNA gel, aerosol. No, that is not possible. You have to have sufficient amount of DNA in order to check that, analyze that. That's why we are doing this polymerase chain reaction. So as a result of that, the answer will be the gel electrophoresis, because you started with a little bit of DNA sample, right? If the reaction is not happening, the control tube, just omit, don't add the Taq polymerase. Everything you added without the Taq polymerase. Now you run sample from both the tube. Everything treated similarly, only in one tube you didn't add Taq polymerase. So as a result of that, where you didn't add Taq polymerase, you will not see any band under UV light. Why? Because the initial DNA is so less that you cannot see, and you added DNTP as raw material. The nucleotide, those are even smaller. Nucleotide, you cannot separate in the DNA gel. It needs to be like at least like 50 nucleotide long, 100 nucleotide long, 1000 nucleotide long, so that you can see. So for example, if your DNA, which you are actually amplifying, if it is, for example, 300 nucleotide long, so you added a lot of DNTP, but that you cannot see in the gel, because I told you have to have double-stranded DNA so that ethidium bromide can be intercalated, that you can visualize in UV. So as a result of that, in the positive reaction, where reaction happened, that means I told you that from just one DNA molecule, how many DNA you are getting after 30-40 cycle. You have huge number of DNA. So although you are carrying out the reaction, say in 50 microliter, just even add 1 microliter solution after the reaction, you will see a bright band there, because the reaction is successful. After 30 reaction, 30 cycle, you have huge number of DNA. Even just 1 microliter, you might have huge number of DNA, and that will ensure that your polymerase chain reaction happen properly. I hope that you understand this part. This is the in combination with DNA, like PCR reaction, and as well as jelly electrophoresis together. This is very powerful technique and very commonly used in forensic science in the past decade, and now also it's very important. In addition to fingerprint forensic and other all those things, this biology or DNA testing, it is also important, and this is routine job in forensic science as well as in laboratory also. Like in academic research lab, it is an absolute, like I would say, indispensable technique. Actually, we are using almost every day in order to clone a gene. Like particularly if I am interested on some gene or some protein, then I have to work with that particular DNA or particular gene. So as a result of that, I need to amplify that DNA, and then so as a result of that, this is a very important technique, and we are employing in a routine basis. That's all. And the next topic is DNA sequencing. That after getting, after amplifying this DNA, for example, as I started the class, that in a particular forensic setup, if you have a little bit of DNA extracted from some cells. So for example, in a crime scene, that investigation team, they collected some hair sample, like just hair. And at the bottom of the hair, you have some hair follicle, like some whitish portion, like some live cells are still there. So even just from few hairs, from the hair follicles, it is possible to extract the DNA, right? Now, you have very little amount of DNA. Neither you can run a gel to see the DNA, nor you can sequence it. You need little higher amount of DNA in order to do that. So as a result of that, first thing will be you have to amplify this DNA. How you will do that? You have to employ this PCR technique so that you can amplify this DNA. Now you have higher amount of DNA. Now how you will investigate this case? The investigation team, like they want to find out the convict or analyze this thing. So as a result of that, now you have to sequence the DNA. Like all of us, we are human individual. So we have very similar DNA sequence. But if you sequence for a long time, like if you want to read a huge stretch of DNA, you will get some differences definitely present in DNA of like my DNA and your DNA. You will see some differences that you have to establish by means of DNA sequencing. In the next class, I will discuss about this step, the DNA sequencing, how we do DNA sequencing in modern time. I will tell you the rationale behind this technique, the chemical reaction, as well as the improved version of this DNA sequencing, so that very very efficiently now we can sequence our DNA sample. It's kind of a routine job nowadays. So that I'm going to discuss in my next class, DNA sequencing. So all these three together, that DNA gel electrophoresis, that is also called agarose gel electrophoresis, then polymerase chain reaction, and this DNA sequencing, they make all these three together, they are clearly some important techniques we use for the analysis of DNA. That's all. So by then I'm just finishing today's class.